Okay, hello everyone, and welcome to the Oxford seminar. This week we're hearing from Elizabeth, who will be telling us about AI for precision oncology.
Hey, thank you. Thanks for having me. So I'll just start off with the presentation. So it's about AI for precision oncology. And I just wanted to start off with the project AIM, which is to create a machine learning model that can predict a patient's immunotherapy response using transcriptomic data. And that's just like basically a sentence that encompasses everything that's happening, but I'll just break down each part as we go along. So I wanted to start with cancer and what cancer is, how it works, because that's quite fundamental to the particular methods that I'm using. I'll go to this link over here, which should open. Okay. So on here, it has some really good images to just explain. So I'll enlarge this one. And so here we have a cell. And I don't know if this, I think the pointer does lag a little bit. So here we have basically a cell, and within the cell we have the nucleus, which houses all like the DNA, all of the genetic code, what's used to make up all the little building blocks within our body. And within that we have the chromosome, and then we have our DNA, which the DNA is basically compacted into chromosomes. And this is what the DNA looks like. So we would have long stretches of, we call them nucleotide base pairs, and they're joined together by other molecules as well. And along these long stretches, some of them, a group of them, can be seen as genes. And these genes are really the things which go on to code for other things like proteins and things that the body needs. And I go back here. So how does cancer form? So let's say that this is just the skin, and this would be what normal cells would look like. But with cancer cells would have cells which grow abnormally within that area. So they either stop growing or they proliferate, divide too quickly, and they start to take up nutrients that they're not supposed to, and things like that. So that's what we classify as cancer cells. And the way that this usually happens is that, if you remember from the picture of the cells, so over time, cells are supposed to just replicate, and then the old cells die and the new cells keep on going. And whenever they replicate, they have to replicate every single thing within the cell. So everything in the nucleus, the DNA, everything has to replicate, and then the old cell dies. And there's different machineries there that ensures that when the DNA is replicated, it's replicated correctly, because that's really important. But sometimes there can be mutations that happen during this replication, that the machinery doesn't catch that, oh, this part has been wrong, let's fix it. Okay. Actually, sorry, I should have switched off my notifications.
Here we go.
Yes, so I think I was talking about mutations. So when these mutations happen, and if they happen within the sections of DNA that are genes, the things that encode for things, this is usually why cancer happens. So going on, so this, we call them oncogenes when the original gene has been mutated into something else. And this is like, well, the picture to depict what a cancer cell would look like. So we'd have our normal cells, which have the normal DNA, and then we have the cancer cells, which have the mutated DNA. And we have something called tumor suppressor genes within normal cells. So these are the genes that basically stop a normal cell from either going on past its lifespan or dividing and the word is proliferating more than it should. And when these when there's mutations within these tumor suppressor genes, that can also cause normal cells to become cancer cells. And so here we show what a tumor looks like again. So this, you'd have a tumor, and then this would also be called the tumor microenvironment. So just the environment around the tumor that enables it to continue growing and proliferating the way it needs to. And it usually calls in a whole host of different cells to either protect it or to help it keep being a cancerous cell. And then we would ask the question of why doesn't the immune system come and just fight against these particular cells? But over time, cancer cells have learned ways to evade the immune system, whereby they'll either act like their normal cells, present things called ligands or antigens on their surface, which show that they're a normal cell, and the immune system won't see them. And they also have other ways that they evade this too. And within like each individual person, usually their mutations will be in different places. So you could have the same therapy, let's say immunotherapy or chemotherapy, and you'd give it to two different patients, but then one of them will respond and another one won't respond. And it may be because of those different changes within their DNA, or even because of the tumor microenvironment and what's happening within that specific environment going on. And then what causes these genetic changes? It can be like a whole host of different things, from environmental causes, lifestyle, cell division, or even like hereditary changes, or hereditary causes like things that go down your lineage. And also age can factor in a lot with cancer and who gets cancer, because as you said, throughout your lifespan, your cells are always dividing, always replicating. And when you're younger, there'll be fewer of these little mistakes that are left during the replication. But as you grow older, these mistakes can accumulate and lead to cancer. And what is metastasis? So with cancer, we have benign cancers, which so cancer, a tumor which is in its original place. So let's say it's in your colon or large intestines or your foot or your skin, and it's just in one place. And these cancers, you can usually just go in with surgery and remove it. However, something can happen called metastasis. This is where cells from the cancer go on into the bloodstream or into the lymphatic system, and they go to other places within your body, and they begin to create a tumor there. So you'd have maybe your original tumor down here, and then you'd have your metastatic tumor maybe in your lungs. And this makes it harder to fight against the cancer. And this is usually when you'd have something like chemotherapy or immunotherapy. So going back to the slides. Yes. So going on to speak about cancer immunotherapy, which is the treatment that I mostly look at. I'm looking at two types of immunotherapy called anti-PD-1 and anti-CTLA-4. And what immunotherapy does is it leverages the body's immune system to combat the cancer. And the way it does this is that we would have a tumor cell here, and then we'd have a T cell. So the T cells are the immune cells. And what the tumor cells would do is they would start to present these ligands over here, or these antigens, which will say, I'm a normal cell, don't kill me. And then the T cells will come by and they'll be like, okay, this is a normal cell, we'll leave it to grow. And what anti-PD-1 does is it covers off these ligands so that when the T cells come, they will realize that this is actually a tumor cell and they'll go on and destroy it. And anti-CTLA-4 works in the same manner, but just on a different ligand. So the anti-PD-1 works mostly around the tumor microenvironment, whereas anti-CTLA-4 works more within the bone marrow. So those are the only like small differences between them. So what is the problem? What's spurred on this research question? So it's that 20 to 40% of people respond to immunotherapy. And we've seen that within those 20 to 40% of people, they usually go on to live longer, more than 10 plus years after receiving the therapy. But on average, well, in the US, it costs around £150,000 for one patient's therapy. In the UK, it's around 100,000. But we don't know how to ascertain which patients will respond to this treatment. And this treatment is usually given to patients whose cancer is in the late stages or they have metastatic cancer. And we just want to make sure that the patients who won't receive relief from immunotherapy don't have to go through the different side effects of taking immunotherapy or even the time that they'll waste trying a new treatment that won't work for them. So at the moment, the current solutions which don't include artificial intelligence is precision oncology, where they use different genetic profiling and biomarkers, and I'll explain more about what biomarkers are, to just find out more about that specific patient's cancer and to personalize the treatment to them. And the current biomarkers they have is something called tumor mutational burden. So remember how I was speaking about how different mutations within your genes can lead to cancer. There's a way to ascertain how many mutations have happened and whether or not this can lead to a patient being a responder or not. And then PD-L1, if I go back here. So PD-L1 is this antigen here that's presented on the tumor cells. And they found that some patients who have more PD-L1 expression will respond to immunotherapy rather than those who don't. But the current issues with these biomarkers is that they can be costly to ascertain, like all of like what's the tumor mutational burden, what's the PD-L1 expression, and the results can differ across different hospitals, establishments, and labs. So there's no universal generalizable way to know that, okay, these patients will respond or not. So going on, my proposed solution is to include machine learning with precision oncology, because another issue with precision oncology is that there is a vast amount of data. Even with something like, I think that's getting ahead of myself, but I'll explain more. So, okay, so I use mostly something called transcriptomics. And what transcriptomics is looking at RNA sequence data, gene expression data. So this is like the genome, the DNA of a patient or any person. And what would happen is that when a certain protein, the body needs a certain protein or a certain hormone or something, it will signal to the cells that we need this much of this particular protein. And there's some machinery that will go to the specific part of DNA that is needed, and it will replicate that part because that particular part is what's needed to kind of like a computer code to enter into something else and say, give us this much of this much gene. So it will replicate that much of it. And that's what we call the transcriptomics, the RNA. And what happens to the RNA after that is that you'd have another machinery that will come and it will translate that RNA into proteins. And so then you have the proteomics. So I'm in this middle area here with the transcriptomics, the RNA sequencing. And what I look at particularly is the transcriptomics of the patient before they receive the immunotherapy. So the data that I have, see if I have it, I go to, yeah, so I'll go to this slide first. So the data that I have is basically, this would have to make this unidentifiable, like, what's the word? Anonymous data. And then you'd have the response. So would have the data of whether or not the patient responded to the therapy. And then this here, these are the different genes. And what would happen is a patient would come in, they'll have a biopsy, they'll take out a part of the tumor, and then that biopsy is what they'll take into the lab, and they'll sequence the transcriptomics from there. So they would want to see with this patient before they receive the treatment, what RNA was the tumor trying to sequence at that particular moment. So that's what this data here is the genes. And then this is all of the expression data. So it was, this patient had this much of this gene. So that's what the data is. I go back here. Yeah, so that's what my data is. And then a biomarker. To explain what more of what a biomarker is, because I am particularly looking for biomarkers, and my biomarkers are either single genes or groups of genes together. And so a biomarker is a molecular or biological, it says here, or physical characteristic that indicates a specific physiological state. And for me, that means it's a gene or a group of genes that can be used to say whether or not a patient will respond. And then you'll ask the question of why don't you just use all of the information that comes in from the patient? But the issue with that is that with each, let's say, data set that comes in, when they sequence the patient's transcriptomics, there could be about 60,000 genes that have been sequenced, and all 60,000 of them are not going to be important to tell us whether or not a patient will respond. So we have to be able to ascertain which ones are actually the important ones, and these are the biomarkers, because in the future, we would like to have a patient come in and we sequence these specific genes, and they're like, and then after that, we're like, okay, you will respond or you won't respond, depending on the gene expression that we see there. And there's many different biomarkers that you can have within the cancer process. So we have here prognostic biomarkers. So we have biomarkers which say how likely a patient is to get cancer. Diagnostic biomarkers that say what type of cancer it is. Then I'm mostly looking at predictive biomarkers, which help to get the optimal drug for the patient. So going on. Okay, so on to the machine learning aspect of this. So my machine learning pipelines are made with something called supervised machine learning. And the difference between supervised and unsupervised is that with my data, I will know the patient response. That's the information that I feed into the pipeline will have the patient response. So the model will find patterns according to what the response is, or try to find patterns that help to show the response. With unsupervised learning, you're giving the model data and asking it to find its own patterns and what it believes is important. And so with my pipeline, the first thing we have is data collection. Data collection is the hardest part by far, because this type of data isn't readily available in many places, because it's usually clinical trials data or really, really old data that's stored in very interesting ways. So that's the first part of it, just collecting as much data as you can. And another problem we have is that you may get a data set with 60,000 genes, but you have like 20 patients. And that type of high dimensional feature space doesn't really help with machine learning, because if you just have like 10 samples and like 60,000 things said about those 10 samples, the machine, the model doesn't always understand which ones are important or not. It can just pick ones which it believes, right, this one's kind of high enough to like say something. And so after data collection, we have pre-processing. Pre-processing is another very important step, because every lab and institute stores and collects their data differently, and you have to be able to standardize your data properly before you use it to ensure that you get the best out of it. And then after that, you have the model selection and training, which is where you'd pick your algorithms and your feature selection methods. And then after that, you have to evaluate your model to ensure that it's actually predicting what it's supposed to predict. And after that, you can deploy your model to use with other external data sets. Okay, so my data is transcriptomic data from pre-treatment tumor biopsies. It's collected through literature reviews, public databases, and clinical trials. And we use the pre-treatment tumor because it provides a baseline of the patient's disease before they receive the treatment. Okay, and then this is a table showing different data sets that I have used. And this here is just like code names for the different data sets. So it's like immunotherapy 1, immunotherapy 14. And these are just the papers that they came from. And the cancer types, they're all from patients who have melanoma. My biggest data set at the moment is 121 patients. And here it says which drug they received, whether it was nivolumab or pembrolizumab. These are both anti-PD-1. And then here are the number of genes in common. So for these different data sets, so they all had their own specific feature spaces or specific genes which were sequenced for them. But in order for me to compare them against each other, they all have to have the same genes. So they all started from around 60,000, 50,000 and brought them down to 7,000. And then with drug response, the way that doctors ascertain or clinicians ascertain that a patient is responding is they use something called RACIST, and it has different categories, and it works with imaging data of the tumor size. So for a patient to have received complete response is their tumor was a certain size and now it's completely gone. So that's what CR is. And then partial response is the patient's tumor was this size, but it shrunk. And then SD would be stable disease. So the patient's tumor hasn't changed at all. And progressive disease is the patient's tumor has continued to grow. And we just have the number of patients here. And for me to ascertain responders and non-responders for this particular experiment, I decided to put complete responders and partial responders as the responders, and the non-responders as people who have sustained disease and then progressive disease. But in other experiments, I could have put them as complete response, partial response, sustained disease as all the responders, and then progressive disease as just the non-responders. And another issue that we have is that at times data sets can be quite imbalanced. So this one here is quite balanced because you have 19 patients who are responders, 22 who are non-responders. But then with a data set like this, where you have 11 patients as responders and then 45 as non-responders, usually the algorithms and the models, they don't have good ways to make sure that they're actually not just favoring the class which has the most amount of people in them, and they're actually finding out those patterns and predicting properly. So they're just like many different problems together that you have to find solutions for as you go along. So go to the first step, which is the data pre-processing. So what we do a lot of is data normalization. And this is because if we go back here, so even with like this data set here, we can have one gene where the numbers range from 1 to 10, and then another gene where they range from like 3,000 and something to 33,000 and something. And we're not going to be able to compare these genes against each other if they have such large and varied scales, which is why we normalize them. And we mostly use something called Z-score normalization. There's many different other normalization methods as well that you can use either in combination or separately. But this one is quite good in that it brings everything to the same scale. Then after normalization, you'd handle the imbalanced data sets and use something called resampling techniques. And there's many different ones, but this is quite a simple one. And most of the time in machine learning, the simple things are the better things. Complexity doesn't always help. So you can either use undersampling or oversampling. Undersampling is where you'd undersample the majority class, but I mostly use oversampling because with undersampling, you could lose information that was actually quite important. So I tend to just oversample the minority class when I am creating these data sets.
I don't know what the majority and minority class is.
Okay, so I go back here. So here, let's take this one, the I-14. So the responders would be the minority class, so they have 11 patients, and the non-responders have 45. So with oversampling, what I would do is when I'm creating my data set that I'm going to feed into the model, I would take all 45 of the non-responders, and then from the responders, I would have a different algorithm that would basically pick from the 11 until I have 45, like randomly.
Sort of like build up the responder class to be of the same size as the non-responder by like duplicating some of the, okay.
Just at random, just to make sure that they have an equal number.
Is it like literal duplications of that data, or do you like do some random noise or something? Or is it...
Oh, no. We don't want to introduce noise.
Okay.
We don't want any noise.
Whereas undersampling would be pruning off the non-responders or something.
You just pick some of them at random, which isn't always the best way because you'd lose a lot of quite important data that way.
Right. So maybe explain why, but like what goes wrong if you don't have like equal size data sets?
So if you don't have the equal size data sets, you could just put it into your model, and then what the model will find is that most of the patients are non-responders. So at the end of it, it will just predict most of them as non-responders. That will be the pattern that it finds.
Right. Okay. It's not going to be penalized enough for getting the responders incorrect because there are so few or something.
Exactly. Yeah. It won't look too deeply into the data. It will just look at that particular relationship and then make predictions on that relationship. Yeah. Whereas we would want it to sort of like look past that and then look for other things within the data too. So going on to the model training. Okay, so for this one, nested cross-validation. Okay, if I lose you on this one, just let me know. I think this one took me a while to understand when I started. So nested cross-validation. Cross-validation is what we use to ensure that the model is actually predicting correctly. It's a way both to tune something we call hyperparameters. So with each algorithm and feature selection method that we use, they all have their own individual hyperparameters that need tuning, which is, let's say, different metrics within each of them that help them to make their specific decisions on prediction. So we could have like, maybe I should have explained the okay, I'll explain this slide first, the hyperparameter tuning slide. So let's say that these purple dots here are all of our different samples, and we're trying to create we're basically trying to draw this line that helps us predict what each one of these circles are. So let's say we have the X for all of the different circles, but we're trying to find their Y. And we want to draw a line that can help us to predict each circle's Y. And we have something that's called underfitting. So when your model is too simple and you just draws a line through the data, and it can predict some of them correctly, but most of them will predict them wrong. Then we have the optimal line that would be drawn, whereby it predicts most of them correctly. Then we have something called overfitting, where the model is so complex that it just tries to capture everything, including the noise, the outliers, and everything that's going on. And the way that we get to this optimal setting is through something called hyperparameter tuning, whereby we would have, let's say, what's that? I don't know what that, parabola. Let's say we have like a parabola and we're just changing like all of the different coefficients. We're just changing all the different coefficients at random to see which coefficients are best to draw this smooth curve. That's what the hyperparameters are.
Okay, so is like the hyper.
So I'll explain more about what each one of them are. Like
in this picture, you could imagine your hyperparameter is like degree of my polynomial.
Right. So the idea that like the parameters are like the weights in the model, the hyperparameters are the like parameters of the algorithm that produces the model or tree. Okay. Right. So like in particular, what the architecture is, how many nodes you have.
Oh yeah. Those are the hyperparameters. Yeah.
That's a line, that's a curve, that's a really high degree of polynomial, right?
Oh, I see.
So that's what I'm just in this picture.
Right.
Like,
Just looking at the pictures.
Yeah.
Exactly. So in this case, it's like simplicity of the model, which is the hyperparameter that you're selecting for. Okay.
Yeah. Yeah, if I go back here. Yeah, so that's what nested cross-validation is usually for, to do that hyperparameter tuning and that feature selection, and also to evaluate how well the model is doing. So when we're doing machine learning, we have what's called a training set and a test set. And the training set is what we would train the model on. And then the test set is something that we would hold to the side, so that after we've trained the model, we can then bring in this data that the model hasn't seen before and have it make predictions on that data to see how well it performs. So if I go back here, so let's say we have this data set here. What we would do is maybe go up to patient 17 and we'll say, okay, that's our training data. And then the rest of this would be our testing data. And during the training process, the model isn't allowed to see the testing data, so that afterwards we can see if the model really understood the patterns within the training data to be able to make these predictions. So when we're testing it, we'll just give it the information on the genes. We wouldn't give it the response, and then it would predict what responses it believes are for those particular patients, and then we would compare against them to see how well it did. So with the nested k-fold cross-validation, K is just how many folds you decide to do. So with this one here, we can see it as a 3-fold cross-validation because we would have like three folds. So we'll split the data into 3 different folds. We'll hold away 1 fold, the light green fold, as like the testing part. And then we'll go into the other 2 folds which are left, and would again split that data into 4 folds, which is why it's a nested cross-validation. So we split that data again, and we have another training and testing part. And within those 4 folds, we would put in the data, would have the model train on that data, and then test on the gray fold. Within that nested, we call it the inner cross-validation. And what it would do is that in those 4 times, what we want to ensure is that we don't assume the model works properly because it split itself in a certain way that was advantageous to it. So we want it to make sure it trains different times on all the particular data within all the folds. And while it's going through those different iterations, it will pick different hyperparameters and different features that it believes are important in each one of those folds. And after that, it would come up with like an evaluation metric. And these evaluation metrics, I mostly use MCC, which is the Matthews correlation coefficient, which I'll explain more about later on. And Matthews correlation coefficient goes from -1 to 1, where -1 means that it predicts everything negatively or inversely. So everyone it believes should be a responder is actually a non-responder, and vice versa. 0 means that it's as good as random, and then 1 means that it has perfect prediction. So within those 4 inner folds, it would have come up with these are the hyperparameters I think are best. These are the features I think are best. And then this is my evaluation metric. This is how well I did. And at the end, we'll pick the one which has the highest evaluation metric. And then that's the one that we would use to train this entire fold and test it on this data that we've been holding out to the side. And then we'll do that a couple of times. So in this case, you do that 3 times, whereby you'd have 3 different sort of like offerings of like, these are the hyperparameters which are amazing, these are the features which are amazing. And then again, it will pick which one is the best. And then that's the one, that's basically your model. That will be like, okay, so now my model is this algorithm with these hyperparameters, with these features. And that's the model you would then deploy to test on outside data, that data the model has never seen before.
So even here, there's some amount, I'm guessing, of like, so here it's like, you pick like 3, 4 for like your folds, and like finding out which are the right numbers to pick. I'm guessing like if they're too high, then first of all, it takes longer to train, but also it might like overfit some stuff. So it's like also a game of like just trying different ones and seeing which ones, okay.
Yeah. Which that was like one of my biggest issues to start with, because usually you'd want to ensure that you'd give the model basically as much time and as much division of data so that it can make as informed a decision as possible. But that's what leads to overfitting. So I wanted to start with like, 10-fold cross-validation.
Yeah.
But again, my data sets are small. So if you're splitting like 40 patients across 10 folds, and that's 4 in like, so it doesn't really work. So you always have to try and figure out like, what's the trade-off of like, do I go smaller, do I go larger, and things like that. Then, yeah.
Sorry, little question. Sorry. So the hyperparameter tuning, that's what happens in the inner resampling, is that right? And then you pick the best one.
Yeah.
The where does the feature selection happen?
Yeah, that happens in the inner resampling as well.
Oh, okay.
Yeah, so that's where it happens as well. So you have to make sure that everything happens within that inner fold and it never sees that test set outside. Otherwise, we get something called data leakage, whereby the model sort of peaks at the answers, and so it makes its hyperparameters specifically tuned to the answers. And that might give you like better results of like, oh, my model's amazing. But then if you bring in another data set that it's never seen before, it's not going to perform that well.
With your, I don't know, maybe you'll talk about this later, so let me know if you are, but like, do you ever use like the other data? So you showed that table of like 4 columns of data sets you have. Do you ever test models trained on one on the other data sets, or do you only test them like internally to that data set?
Yeah, so that's the thing that's what we want. In a perfect world, we want to be able to take one data set from like one hospital, one institution, train a model on it, and then another institution in another country bring that data and it'll be able to predict it. But that's the biggest problem so far, the generalizability of like training a model on one specific data set and then it working on another data set, even like within the same country, it's still really, really hard to do.
Does this just come down to like different labs use different tools, like have different, like it's this kind of level of like?
Even like where the tumor was, like the tumor biopsy site, many different things could like go into that specific question of like, why don't these genes work with this other data set, which is, that's actually like the main question of my thesis. Yeah, so going on, okay, feature selection. So feature selection, I'd say is my most, that's the part that I'm most excited about, because this is where you pick out the specific genes that the model believes is important for making these decisions. And we have different methods that we use, and we do it because of like the high dimensionality of the feature space, and all of the genes, all 60,000 of them are not going to be important. Can we find like maybe 10, 25 of them which are important? And it's a hard thing to do because, for one thing, most of the feature selection methods, they tend to focus, look at them per gene. So like what ANOVA does is it looks at, okay, if I go to the next slide, yeah. What ANOVA does is it looks at a particular gene across all the samples, and it looks at the variance between the responders and non-responders. And if there's a high variance, it will say, okay, this must be an important gene because the responders had this expression, but the non-responders had this expression. And that's what ANOVA will do. But genes and the body don't work that way. They don't work individually as one by themselves. They tend to work together, either in pairs or not even like in pairs, in groups together, or like they're working against each other and things like that. So there's other methods to try and ascertain these relationships, like mutual information. So mutual information is more pairwise, and it looks at two genes together, and it says, okay, if this gene is gone, what information do we lose in making the prediction? But if this gene is gone, what information do we lose in making the prediction? And it gives them all a score. So the ones with the highest mutual information are the ones which it believes are important for the model to make these predictions. So that one it's better than ANOVA because it looks at relationships between genes, but it's still just looking at two genes together. And then another one, LASSO. So this one is more of like a regularization technique, but it can be used for feature selection. And whereas the other two, ANOVA and mutual information, how they would work is along your pipeline, after you've done your data pre-processing, you've normalized your data, you would then use the feature selection to pick the genes which are important, and then you'd go on to train your model. But how LASSO works is that it picks the genes while the model is training. So it's ascertaining which ones are important while the algorithm is also training the model. And it helps to cut down how many features you have, in that it applies a importance score to all of the genes, and then it applies a penalty to every single gene. And all of the genes which don't go down to zero are basically what it deems as the most important ones and the ones that it keeps.
So they're like pruning the inputs as it's
Yeah.
training them. Okay.
Yeah. So, and then these were just like graphs of like different genes which had been picked. And then going on to speak about the algorithms, I actually thought about putting all the maths up here, but I can't explain the maths. I just left that out. So yeah, there's different algorithms that can be used. I like to use a wide selection because they all have different types of information that they can give you. So with the support vector machine, it can work with finding both linear and non-linear patterns and relationship within the data. And as you can see from the first one, this is whereby it would work in a linear way. What it does is it creates something called a hyperplane, which is a separation between the two pieces of data. And it can have different kernels, like this one's called the linear kernel, the polynomial kernel. I think this one's the Gaussian kernel, whereby it will have different ways to create that hyperplane. And this one, and it always creates the hyperplane to sort of like call it the decision boundary to separate, in my case, the responders and non-responders. And it does quite well for the type of high dimensionality data that I'm looking at. So it's a good one for me to use. And its different hyperparameters are C and the kernel. So you can pick which kernel you want to use. So it's the linear kernel, the polynomial kernel, or the Gaussian kernel. Going on. So logistic regression. So what logistic regression does is it maps all of the data onto sort of this XY curve, and then it draws this sigmoidal curve, and then you have to pick the threshold value of, okay, for here they chose a threshold value of 0.5. So everything above 0.5 is a responder. Everything below is a non-responder, but you could change this threshold value and tune it so that it can it can separate the data in different ways. So that's another hyperparameter that you could use. And then, yeah, decision trees, which I think everyone here is very well acquainted with. Yeah, you have your decision node, and you just the model will keep asking itself different questions of like where to put certain genes, where to put certain samples according to like the gene expression values. And at the end, it will decide if a patient is a responder or non-responder. Hyperparameters will be like how deep you want the tree, like how long you want it to go on for, and things like that. And then a random forest is a group of decision trees. And at the end, they'll basically just have a majority vote. So say this many trees decided that this person's a responder, and then we'll be like, okay, so that person's a responder. And then we have another one called extreme gradient boost, which is a group of shallower trees. So you'd have decision trees, but would say maybe their max depth is like 2. And with like each tree learns from the mistakes of the previous tree. And they just try to make the trees better and better as they keep going on. And this you can ask it to do like an iteration of like 1,000 and it will just keep on going, keep on going. So this is one of the more complex models, which isn't always the best, but sometimes it does really help you find those different patterns. So this, we can see it. So this is really like the information that I really care about at the end, which is what genes were chosen by each of the different methods. So here on the side, we have ANOVA, feature selection, mutual information, LASSO, and then at the bottom here, we have some of the genes. Some of the genes from my data set. And the ones just mean that this method chose this particular gene to be important. And you can see that each of them tend to favor different ones for different reasons. But I want to mostly look at the ones that they have a consensus on, like we both thought.
Vertical slices kind of thing.
Yeah. We both thought that this is important, or the 3 of us thought that this is important, because those are the genes which are usually the most important. Because the worst thing is training a model and then figuring out at the end that it was just assimilating to all of the noise within the data set. It never actually found out anything of use, and all the genes that have been picked are actually not important. Which is where like domain knowledge comes in at the end, because after you have like your list of genes which the machine learning model has said are important, you have to go back and do the research of finding out, okay, what does this gene do? What does this gene do? What does that gene do? And that's where clinical validation comes in afterwards. So after you have like your list of genes that you think are important, you can then go to a clinician, maybe someone who works with like animal or cell models, and be like, okay, I found that these genes are important. Can you actually do some experiments to see if you increase or decrease this gene, will this cancerous cell respond or not to the therapy? And that's just another step of validation that helps in that process of picking out those particular genes which are important and then bringing that to a clinical setting whereby that can be used by doctors to make those decisions.
So how does it increase or decrease that gene work?
Like, go back here. To transcriptomics. So within, let's say, like your tumor microenvironment, or within your tumor cell, it can say, we want 1,000 copies of this one gene, and then it'll produce that much of this gene because it wants it to produce that much of this protein. Then they say, okay, we just want 2 copies of this other gene. And you'll find that in a tumor cell that has this amount of that particular gene, the patient will respond or the patient won't respond.
Can you trigger the production of certain genes to like promote the effectiveness of a particular? Okay.
It goes into like genetic editing and all of that other, because you can add like certain promoters and stuff to like different areas of the gene, and then the machinery, I think it shows like the machinery here a little bit. The machinery will go in and actually replicate that part.
I guess it's like independent of the actual immunotherapy. You're just modifying the microenvironment to try and get it to behave in a certain way.
Okay, so it's like it does like 2 processes. So one is this like predictive power of like just looking at what the gene sequence is right now, how might they respond? But then it also opens the door to like these questions of like, would modifying the amount of these genes being produced also have an effect or not, kind of thing.
Yeah, and that would mostly be in the validation side of it. It could work in a way that they'll then go on to maybe do some sort of gene therapy for patients, whereby they do that to make the patient a responder, which is actually something that's quite interesting. Yes, okay, evaluation metrics. So this is what we call a confusion diagram, confusion metric, confusion metrics, confusion matrix. There's the word, confusion matrix. Yeah, I look at these all day long. But yeah, so this would sort of be like, I don't know if you have these in maths, but you'd have let's call, yeah, so observed is what it actually is, predicted is what our model predicted it to be. Can call species K the responders, other species the non-responders. So true positives are all of the patients who are actually responders and were predicted as responders. And then false negatives are the patients who are responders, but they were predicted as non-responders. And then the false positives are all the patients who are non-responders, predicted as responders. And true negatives are those who are non-responders, who were predicted as non-responders. And that gives us all of these evaluation metrics. Like accuracy. Accuracy isn't applicable in what I do because of how imbalanced the data sets are. Because it could turn out that if we go back to the example of I have 11 responders and 45 non-responders, if it just predicts most of them as non-responders, the accuracy is going to be high and we'll be like, oh, this is an amazing model, but we don't actually know if it's an amazing model. And so what specificity does is it looks at all of the non-responders who were actually predicted as non-responders. And then what recall would do would look at all of the responders who were predicted as actually responders. And then what precision would do is it would look at the number of because it's quite hard to like explain the difference between precision and recall. So precision looks at all of the responders who were actually predicted. So how many of the predictions that were made to be responders are actually responders? Does that make sense? Yeah, so it's basically looking at how good is the model, whereas recall is looking at how many of the people were actually predicted correctly, if that makes sense.
Yeah.
And this is always like a trade-off of like, how good do we want the specificity to be? How good do we want the recall to be? Because you'd almost say that you want the recall to be the highest because you want to make sure you get all of those responders. But if you're doing that at the expense of specificity, then you're still in the same problem because you're still giving a large amount of people a drug that most of them don't need. Whereas if then you'd say, okay, let's trade off the recall a bit, then you'll lose patients who would actually respond to this therapy are now losing out to it. So it's always hard to sort of like pick which one you care about the most and then go back and tune your hyperparameters in accordance to this. And MCC, Matthew Correlation Coefficient, is the metric that we use the most because it takes into account everything within the confusion matrix. Whereas accuracy would take into account some of them, MCC would take into account everything. And from the MCC, you can actually see whether or not you have a good model. Because if your answer is around 0.3 to 0.5, that means that your data and your model has a relatively good predictive performance. Getting an MCC of like 0.8 is like, this is amazing, put it in the hospitals right now. Anything around 0 is like, yeah, this isn't really working. Okay, so I wanted to put in here some of my data to show like how, like to just show some live results. So this is my best performing data set, the IO9 data set. And it's made me like ask questions of like, why do you perform really well? Why don't the others perform really well? Because here we can see that when we use logistic regression and with lasso, well, lasso does seem to perform quite well, then you'd get an MCC of like 0.675. But here, when you use the classification regression trees, the MCC is always quite low. which we can see that as, because we're using quite a simple model, just decision tree, then that's why we would create models that don't work that well. Whereas logistic regression and support vector machines do seem to work quite well with this data and random forest. And then we could say that the XGBoost is probably then like on the other side of things, too complex. Basically too complex of a model to use with the particular data, because the data is around 52 patients. So XGB might be too robust for that number of patients. And so after I got all of this data, and I then picked out the specific models from each of these. So we would have a model that is like CART and ANOVA, and then that would be like a particular model. And then another model would be like logistic regression and ANOVA, that would be like another model that could be used. I went and tested it on another data set, the I-15 data set. And you can see here that the MCC is not that good, it's all around 0. So even though so this means the CV cross-validation MCC. So this is the same as this one. This was the cross-validation MCC of the IO9 data set. So even though with like logistic regression plus LASSO, we got CV MCC of 0.69, when I then brought in another data set, collected in a different lab, different group of patients, the MCC was not that good. Same with SBM and LASSO, which brings in the next question of how do we make these models more generalizable? Like, what is the specific thing that we could do to help with that?
Do you have a sense of whether it's because like there's just some incompatibility between the data that they present or the methods they use to get it, or is it like maybe you just is cross-validation like or is the way you produce this 0.69 number like how like reliable is that as a metric for the effectiveness of this model?
So it's a metric that shows the effectiveness of that model for that particular data set, for just that data set. When you bring in like an outside data set, then it would have a different evaluation metric. And through like more experimentation, what I found is that, well, first of all, using just one feature selection method to pick how many genes were important isn't useful, because they will all pick ones which they believe to themselves are important. But if you sort of bring them in to have a consensus of like, who did all of you believe is important, which isn't data that I put into these slides, then they come up with a more coherent list of genes which they think are important. And then you can go on to do other things to sort of look at, I think I have them in here. So I had like extra slides. Is that, yeah, so something called ice plots. I had some, no, there's no pretty ones in here. So yeah, so we have something called ice plots, which you use to ascertain how a gene works. So let's say one of them chose, this is a very long, NAP1 as an important gene. We can then use an ice plot to see and have a look at why does it think it's important. And what an ice plot will do is it will get the expression of the gene, and it will change it, and it will see how the prediction of whether a patient is a responder or non-responder will change. And with those specific ice plots, you can then see that, okay, this model thinks this gene is important because it's saying when the expression for this gene is high, then this patient is more likely to be a non-responder. And sometimes when you do your whole machine learning pipeline and you get the genes and you do the ice plots, some of the ice plots are just flat. I see it.
You're expecting the gradient to be high,
like high, low, just something to happen, but they're just flat. And I can show you that okay, so the model picked it, but it's not actually useful. But when you do the consensus of like, what did ANOVA pick, what did LASSO pick, what did mutual information pick, it gives you a crop of those different specific genes which actually had an impact on the answer. And that's like my continuing research to go on creating these consensus biomarker panels, which carry more information or more important genes that help to make these decisions. And it's interesting because like it helps to reduce the dimensionality a lot for one particular data set. But then if you have like, if I get my three data sets, three different data sets, and I train those different data sets, and then they come up with their own panel of genes, and then I bring those genes together and create a larger consensus panel, I find that it was actually an interesting finding. I found that one particular data set will like 7 of the genes and it'll perform really well with those 7 genes. And then another data set will pick another 2 that it thinks are important. And then another data set will pick like 3. And then this one will pick just 1 that it thinks is important. But it's all from that same panel of genes that they all decided were important. And so it just begs the question of like, why did you pick this particular one? Is there like, is it because of where the biopsy was taken from? Is that particular gene more rampant in that particular area? Or is it because of like the overall makeup of the tumor microenvironment, what's happening in that particular area, that this gene was helping to make the decisions there? Yeah, and it's even like the different functions of the genes, which are interesting as well, because one of them, and go back to this other picture, be helpful. Yes. Oh. Oh, I think it was actually on the, yeah, here. If it goes back to the first, okay. So in this tumor microenvironment, it has, as I said, it has like its own sort of like little mini village town that it's created for itself. And it might, so there's something called fibroblast within the skin tissue, and it's what sort of, I don't know if biologists will kill me for saying this wrong, creates like the scaffolding around within your skin. And sometimes within the tumor microenvironment, it can cause there to be more fibroblast within that particular area so that immune cells just can't come in. And so you might find that that gene is highly expressed and it's important for that particular group of patients because they had high fibroblast within that particular area, and the gene for making fibroblast was expressed more. Or in another one, you would find that the tumor cells needed more blood vessels, so they were calling upon a gene that produces more blood vessels. So for that particular data set, the patients who would be classified as responders or non-responders depended on the gene that produces blood vessels. And it's just like different nuances that they have. And my overall like question is to find out a way to ascertain that, like to get a data set or to even get one patient sample and be able to say, these are the biomarkers which will work for this patient sample. And I have like different experiments which I have created to sort of like go forward and answer that question. Yeah.
Right. Is it going to be like, if like these genes are more important in the early stage of the tumor and these in later, or like what? So are you saying you have extra information about like the patient that tells you which subset of genes should be marked as important?
Yeah, so not even having like just like the information we have at the moment, like or like the transcriptomics of the patient's tumor, can they provide an answer of what biomarkers should be used? because right now they're trying to find just one biomarker and use it with everyone, every single hospital, everywhere, or maybe a group of 10 and just try and find like one model that will work for all, which I don't think that will really work, because well, it's showing us that it's not working and everything's different, everything's complex. But can there be a way for the patient sample or the sample group to tell us which biomarkers would be the best to use to make predictions?
That's really, so like, yeah, is there a trade-off between, like, so you showed, for example, when you like do the cross-validation MCC, like something that scores really high, it gets 0.69 or whatever, scores then around 0 on something else. Is there a tendency that like things with a less impressive cross-validation, so say it's like 0.3 or something, perform more consistently across other data? Like, do you notice this kind of observation that like the MCC is sort of like a specificity to that data set being good, and lower MCC means they might perform better across other ones? Or is it generally, no matter what the MCC is, the moment you test it on another data set, it's kind of 0?
Yeah, so that is actually something that I found with one data set in particular. It was a data set that I was fighting with for a long time, because it kept, whenever I'd like train it, so you see how the data set I just showed, it was showing like MCC of 0.9, 0.3. Whenever I train it, it would get like an MCC of 0, 0.1, 0 point something. And I just had to use like different normalization techniques, different algorithms, different processes to find a way to capture basically the signal and patterns within the data, because most of the time we'll just say that that data set isn't really useful and put it to the side. However, there's too many data sets which are not really useful that we should put to the side. So I was just thinking maybe there's a way to like just glean some information from it. And after I had done that, I found that finally got its MCC to around 0.3, 0.4. And when I then create a consensus panel from that one, it did perform generalized better with like other outside data sets. And I don't know if that's like true for all, but that was true for that particular case.
Yeah, that's really interesting. Like this idea of like sort of like measure between your metrics of like not just MCC, but sort of like variance of MCC, trying to minimize this. And then like you would hope that you can also make it on average really high, but like this, variance. Okay, thanks.
Okay. Yeah, and that was it, basically. Yeah, cool.
Okay, shall we? Yes. Okay, we've gone a bit over, but maybe we've got 5 minutes for questions. I mean, we did interrupt you with a few questions.
That's okay.
I have a question.
I have lots, but like,
well, I got a few questions because I just don't know much about cancer and biology, but one maybe more general one is like, it seems like you can come up with a particular model which looks at a particular set of genes, or like particularly looks at the transcriptomics, but maybe like you think that features of the patient's immune system, like later down the line, should be relevant, like for detecting whether they'll have some autoimmune response, or maybe you decide you also want to look at some like genotyping of the actual cancer cells. What methods are there for like taking a model that you might come up with and have trained, and then like using that, extending that to a larger model which can take in more data, or can it like fit in with other models in a compositional way, or do you just have to make a new model from scratch?
There are different ways, calling it multimodal ML models, which is something I like to do, but the issue with that is always that it's hard to get like all of that data for one particular data set. So it's already hard enough to find a data set which has transcriptomics and response. But if you want one with transcriptomics, response, and then tumor mutational burden, they might be saying, oh, you're asking for too much. And then another one would like, you want to add on the PDL1. But in the chance that you'd have like all of the different metrics presented to you, you could train them together from the beginning, or you could build a model on just transcriptomics, just something else, and then you do again the majority vote. Or another thing you could do is fine tune the model. So you'd have like your particular model that you've created and the way it works, but then you'd bring in more data, and I believe it's called transfer learning, whereby you can bring in that additional pile of data and then train the model a bit more. They do it mostly with deep learning. I'm not sure how it happens in the machine learning landscape, but there are ways to do it whereby you already have your model and then you bring in more data to further train the model.
I have some questions, but I'd rather wait until after coffee.
Okay. Do you have any questions? Okay, maybe let's stop the recording there. And let's thank Elizabeth again.